I looked at my records. The last time that I gave a Hanover seminar was in 2009. So what I'm going to do today is give all the talks I should have given in one presentation. Well, not actually, I'm just joking. But really, I have been asked to give a seminar that's more of a retrospective rather than focusing on a specific topic. So what I've tried to do here in this presentation is sort of thread together a bunch of research I've been doing over the years and show you how it all magically connects together into a coherent research program. Okay. So I'm going to start with my approach to forest biometrics. So for those of you who are not familiar, forest biometrics is the application of mathematics and statistics to forestry. It's really started as a decision support science for forestry, and was mainly focused on forest inventory specifically for wood products. Most forest inventories were focused on how much wood is there in the forest to supply human needs. As we're all aware, foresters have a lot more to think about these days. And now we talk in terms of, instead of timber inventory, we talk about comprehensive inventories for ecosystem services. I really like this diagram from the Food and Agricultural Organization of the United Nations. It kind of represents all the modern values that we put on trees. And my field, forest biometrics, has had to expand in order to be able to accommodate all of these different services, or at least contribute more to measurement systems that support them. And a tree is really a good way to understand how I think. I think about trees way more, well, maybe than a man should think about trees, but I think about trees all the time, and sometimes my wife says, "Are you thinking about trees right now?" But for me, if you think about all of these values that are reflected in the needs that people have from the forest, we could sort of think of the forest ecosystems that supply them as being organized into these scales of organization, which we're all familiar with in science. And I kind of am usually operating right around in this zone. And I view forestry, sort of my profession and the department I'm teaching in, as the coupling factor. So people in forestry are linking human needs to the capacity that forest ecosystems can provide. And so in my research, I tend to view forests from a measurement perspective as assemblages of trees, even though that FAO tree shows it's a lot more than that. And I also study trees' behavior, tree form and function from the perspective of different components of trees that make them up. And so the questions that I answer usually come from this side of the diagram. That's usually someone wants better numbers. And my approach, though, because where's Scott? My PhD is in ecology and evolutionary biology, but I trained with a forest biometrician. So I really have this tree biological perspective that informs the way that I do my research. So what I want to have is a unified field theory for trees. Tree models is what I build, and tree models that will be accurate for all types of trees everywhere, all the time. That would be my ultimate model, would be a single model that can be used to describe any tree anywhere that it grows. And another aspect of universal models is compatible estimation of tree components. That means the different components of the trees have to match. You can't have a tree that has too much leaves relative to the amount of sapwood area it has. And then parsimony is using the fewest model possible, because a lot of the agencies that I serve and interact with have to actually go out and apply these models. So we don't want to make them overly complex so that people can actually use them. And the social ecological relevance is really what links these things together. So while I may be designing a timber inventory system for the Michigan DNR, the utilization potential of a tree ultimately, in my mind, stems from what species is it, what's its life history, what's its environment. So This is a diagram I put together, anatomy of a model tree. So you can see the whole tree here, broken down into its core functional parts, the stem, the branches, the roots, and the leaves. And so a lot of forestry or forest biometric modeling of trees has focused on this part, the main stem of the tree, and how do we extract saw and pulp logs and other emergible products. I've been doing a lot of work on the branches of the trees. And because the branches of the trees are sort of like a neglected realm, they're there, they're very difficult to quantify because there's so many of them and they're all twisty and tangly. And so this is a main area of research for me. And again, trying to link the ecology of the tree in these models to the utilization of the tree parts. So what do we do in my lab? We go out and we measure lots and lots of trees. And we mostly are taking dimensional measurements of trees that tell us about space filling of the tree and the volume of the tree, occupancy, if you will. And then we can do more advanced measurements, like maybe taking a core from a tree and then calculating things like density and decay. But if we really want to understand the tree, we have to cut the tree down and take it apart and measure all of its little pieces. How many people remember in high school when you dissected a frog or a worm? So when you take something apart, you really understand it as a living thing. And in all of the experimental designs I put together, I'm always trying to maximize the diversity of species from a functional perspective so that I can truly have data that enable to build generalized models. Okay, in this case, for my big USDA Forest Service biomass project for the Great Lakes, I've been selecting species with different leaf types, different wood densities, and different levels of shade tolerance in this matrix. We do a lot of lab work. So we take parts of the trees back. We do all kinds of chopping, weighing, dissecting, scanning. Here's Garrett Detman collecting leaf scans in Harvard Forest. Here's Sam Clark at Kellogg Forest working very hard, taking notes on all these thousands and thousands of measurements we take on every tree. So what are my key talking points for the rest of this talk? Now that you kind of know who I am, maybe a bit more. My research program serves a lot of different audiences. Today I'm just going to focus on two just to put some bounds on this talk. And my overriding research questions are what new measurements and models can improve understanding of trees, forests, and the human needs that they meet. And then how does tree form and function inform quantification of ecosystem services? So the first segment of this, I'm going to talk about this branch volume estimation work and how it's linked to stem volume. My partner here is the Michigan DNR. The Michigan DNR manages 4 million acres public forest system in Michigan. It contributes tremendously to the economy. DNR foresters every year are going out and putting up tens of thousands of acres of land, public land for timber sales, which is supplying wood to the economy, and the state is receiving considerable revenues from that. They're going to roll out a new inventory system in 2018, I hope, after 13 years of working with them. I've been slowly but surely helping them redesign this system. And the overall goal of my interactions with them had to make the timber inventory system that supports all these services more accurate. They're using quite outdated systems. This is the Lake States volume equations by Gavorkin and Olson, published in 1955. And this is more recent work by Gary Fowler, who was the forest biometrician at University of Michigan when they still had a forestry program. So the key issue here is that a professional forester working for the DNR or a contracted forester has to go up to a tree, look at that tree, and try to assess the wood products that can be extracted from it. However, the trees are diverse in form and products. And so this is where the biological linkage links back to the assessment. So if we look at a tree, we can see that this tree has wood, both saw and pulpwood, in the main stem in blue. And this is just a diagram from their manual, but it has wood and branches. And there are already some pretty good systems in place to estimate volume from this part of the tree, but haven't really been for this branching part. And in some of our trees, we've even found pulp logs and branches on branches and branches, so even as much as a third order branch can contain this wood in a tree with a massive crown. And a lot of this wood simply was not being accounted for. So more destructive sampling. The key things to note here is the destructive sampling. We have professional loggers cut the trees down. These are real timber sales. Then we assess all the timber products can be extracted, and we diagram every tree in a very complex coding system. Things to know about species. We can never have enough data to do this kind of work. You'll see we focused more on the more commercially important species in here. We got almost 2,000 trees, and still we have relatively low sample sizes for a lot of species. So this is one of the main reasons that you need universal models, is because we're never going to have enough data to get the data for every single individual of every species of every type. So you need a good representation of all the different types out there so you can build these universal models. So the underlying model that's most often used is what are called allometric equations. This term kind of gets bandied around and not always used correctly, but basically the original language comes from Julian Huxley, who is the grandson of Thomas Huxley, who was called the bulldog of Darwin. But anyway, so it's not that Huxley. Allometry is the growth of one part of an organism relative to the other. So if you think of my tree model, how are different parts of the tree growing relative to one another? Everybody likes this model, the power law, which came out of this work. And in this case, they were looking at, the original work, they were looking at ants and looking at basically relationships between the size and width of the head and the length. But what everyone really likes about this model is when you log transform it, it becomes a linear model, which is very easy to work with from a statistical perspective. So, taking thousands of trees that we cut down, looking at all the wood volume in them, merchantable wood volume, it looks like it fits this power function pretty well, though you can see there's a lot of scatter. Each letter, two-letter code here is a different species. So we get a lot of variation around this supposed universal model. However, if we plot it in a log-log plot, we see that, in fact, it is nonlinear in a log-log plot. What does that mean? That means as a tree increases in diameter at breast height, the amount of volume is continuing to increase exponentially, but the fraction of that volume that's merchantable is decreasing as a proportion of the total volume of the tree. So if we look at it from a whole tree perspective, we can break it down and look at the stem. So this is also true for the stem, and this is in part because the stem is tapering as the tree increases in size. But I think the real answer has to do with the fact that the trees are becoming more branchy. And so what I really wanted to understand to get these comprehensive volume models for the trees is the relationship between the stem and the branches. So I turned to Leonardo da Vinci, who was the first person who ever really started to try to figure this out. And da Vinci had a rule that said basically the diameters of all branches would add up to the diameter of the stem below the fork. And da Vinci's rule was most famously applied recently in the metabolic scaling theory of West, Brown, and Enquist. This was a major paper in Nature. If you saw last week's presentation, they talked a lot about how this relates to energy and other things. And so they said trees and plants are basically fractals. Actually, that's not true. They're fractal-like. And this is an important reason why this law isn't really universal. Key issue is that the trunk of a tree actually has no fractal dimension. So I fell back to da Vinci and came up with this theoretical idea. way back when, of Da Vinci trees. And so basically the idea is that this is the total volume of the tree, the gray is the main stem, and as the tree branches, wood is diverted from the main stem into branches. And then this trunk length, which is the base of the crown, as it's rising and as the branch becomes smaller and smaller, a smaller and smaller fraction of total wood is found in the branches. And so I created these trees and said this is kind of what a cumulative volume, relative volume profile should look like of these theoretical trees. So I've been working with the now late Dr. Wojtek Jakrzewski, who is the provincial forest modeler for Ontario Ministry of Natural Resources. This related to the Canadian softwood lumber dispute, and we were doing some cross-border comparisons that related to the dispute, which ultimately resulted in the U.S. owing Canada $4 billion under NAFTA, which, well, sorry about that. But this is what's called a taper model or a stem profile. And so this is the diameter relative to dBH. So it's higher down at the flare at the bottom. And then as we walk up the stem gets narrower and narrower and narrower until we get to the top of the tree. So I started looking at this data and thinking, well, I was looking at forked trees and realized that forked trees under this Da Vinci tree model should actually have less volume accumulating in the stem. So you could see that with real tree, and this is when I was still plotting in Excel, so this is an old graph, indeed, it's true. And so then you can make a cumulative volume profile. So basically, if I start at the bottom of the tree and I add up all the volume in the tree till I get to the top, I get all the volume at the top, and then this is at the very, this is the cumulative height above the stump. So this is all the volume. And so you see these cumulative profiles. And so basically, at the base of the, once we get up in the trunk and we start adding branches, we start getting branch volume. So this is the cumulative profile of the main stem, and this is the cumulative profile of the main stem and branches. And so you can fit these to a tree, and the difference between these is the amount of branch volume. So I want to generalize this method, and since Leonardo da Vinci was dead, I brought in Neil Verplank, seen here clipping many leaves off of trees in our biomass study. Neil's here, isn't he? Hey, Neil. Good job here. So Neil's thesis, we basically generalized this model and developed these cumulative volume profiles, and it was a very nice method. However, I was really curious when we started doing this, do real trees look like Da Vinci trees in my original model? And so actually they kind of do. So this line represents the stem and branches, and each of these upper lines represents a divergence depending on where that crown height is and how big the branch is. And so real trees never look exactly like your theoretical models, but overall it was a pretty successful idea. So one of the neat things about this is, so this is again that diameter. This is the relative cross-sectional area of the stem as a function of breast height cross-sectional area stem. So we've got a big wide cross-sectional area down at the flare at the stump, and then it's dropping. Look what happens when we hit that first big branch. We get this drop. So I started thinking, okay, I got to have some simple metric that can capture this. We don't have to build all these complicated models. So I was walking around in Baker Woodlot, and I thought, well, if you could measure the height and the diameter of the largest branch in the tree, that would be a pretty good way of indexing this without having to build these complicated profile models. And so I came up with these two measurements, diameter of the largest branch and the height of the largest branch. And then people were asking me, Dave, how are you going to measure that? So I'd been working in field studies course with a consulting forester who was hawking new forestry tools. And then he showed me the gator eyes. And what the gator eyes do is they project don't lock on me now. They project these two laser beams from a caliper, and so you can measure the diameter of something in any which way or dimension that you can. So now I actually had a way to measure that. We could already measure we already have things like a clinometer or a rangefinder to measure the height to the largest branch. So in this paper, I said, what if we just wanted to calculate the ratio of branch volume to stem volume, because we can get a good estimate of stem volume, multiply that and get the branch volume? What would be a good predictor of that? Well, it turns out that the diameter of the largest branch and the height of the largest branch are very good predictors of that. So much so that compared to diameter at breast height, which is what we were using previously and we use for everything, we get 50% more of the variation explained. If you remember that first picture I showed, there was huge amounts of scatter around the branch volume dBH relationship. That's why if you tell me a tree has a big trunk, you can't tell me that it has a big crown or a small crown. Not enough information. This is a place when I started thinking, wow, this could really be applied to improving allometry of trees for biomass estimation. It's a good method, but too measurement intensive for the DNR. They have to run around and measure all these branches. So I came up with merchantable form types. So I was looking at this diagram and seeing how the heck are we going to come up with models for all these trees? So I said, well, what if we fit a universal model, and then we just assign each tree to a form type, and then look how each tree's form type diverges from the underlying universal model for all trees. And because every DNR forester goes to every tree and basically counts these saw and pulp sticks, I figured we could describe every possible combination with a 16-level code, a binary whether or not it does have pulp in the main stem, saw in the main stem, saw and pulp in the branches, or saw in the branches. So for example, in their manual, tree A is a 1000, pulp in the main stem, nothing else. Tree E here has saw and pulp logs in the main stem and saw and pulp logs in branches. And I teamed up with Aaron Weiskittle. We did some pretty neat mixed effects modeling, which he is quite an expert in. And basically it turns out that if we want to look at all the variation from all the trees of all species and sizes, this is 100% of the variation, that it's much more important to know the merchantable form type than it is to know the species of the tree. So, very powerful classification system. Let me just show you an example. This black line is the universal stem taper model for all trees of all types. And then the way this model works is each tree diverges from the overall trend based on its species and its form type. Now, sugar maple and basswood grow together. Basswood tends to be more branchy. So you see trees of the same form type are above the curve. They have this nice clean stem. Trees of the same form type are more close to each other than members of the same species of different form types. Anyway, this is the winning model, and I hope to have this finally calibrated for the whole state, and then the DNR will use this model moving forward. Okay. The next major segment is building better tree mass models. So I just did a whole ton of work on volume, theoretical work, practical work on measuring tree volume. And at the time I had a PhD student, Zhongli Wang, who was very interested in these big picture ideas about energy metabolism in trees, just over-the-top theoretical stuff. And he got me thinking about energy-mass relations. Well, Zhongli is a really bright guy. But when I want to talk about energy-mass relations, I thought of somebody who is even brighter. Maybe. So Einstein had a lot to say about energy and mass. And so what is mass? Well, mass is energy divided by the speed of light squared. How is that relevant to trees? Well, think about it. What do trees do? Trees decelerate energy through photosynthesis, and they allocate it to mass. What else do we know if we want to work from base principles to build better mass equations? we know that mass is equal to density times volume. Well, I just spent a whole ton of time learning to build better and complex volume equations. So if I could nail the density, then we should have a pretty good generalized model to start with for building tree mass models. Who cares about all this? FIA, the nation's forest census. So basically, the Forest and Inventory Analysis Program is the #1 funder of my research program. And they provide critical information for the management of the nation's forest resources. They put a lot of money into this program, and they do all sorts of things with the data. I pulled up their most recent business report, where I got some of this info too. I looked at their long-term strategic direction. I looked at the top 3 items. The #1 item they listed is to finally go into the interior of Alaska, that scary place, and do inventory there. And actually, MSU Forestry, via the Finley Lab, is playing a major role here. And via me, MSU Forestry is playing a major role here, reporting information on renewable biomass supplies, carbon stocks at the local. So the National Forest Biomass and Carbon Inventory. And I've also been doing work in urban areas. So the National Forest Inventory, until recently, did not cover urban areas. Now, they're extending it, urban FIA, into cities. This is their implementation from 2015 and 2016, and they just completed the first round of urban forest inventory plots in Detroit in summer 2017, so just this past summer. What is FIA? It's a national grid of plots. If you take a grid, a Euclidean grid, and you try to stretch it over a curved surface, the grid crinkles. So at the scale of the United States, the curvature of the Earth matters, so they cleverly use a hexagonal grid like a soccer ball. And then what they do is they divide that into one big hexagon. You could see why Alaska gets ignored. And then they continue to, no, they really have them in Alaska too. And it gets divided into hexagons and hexagons on ad infinitum until we get down to the FIA plot. And the FIA plot, we go, and this is Lisa Parker, for those of you who remember her. We go and measure plots, and then right now they measure DBH, and then they use allometric equations to predict mass. So the key thing we can do with this data is we can scale it up to make these broad scale estimates at the local, regional, state, continental, global scale. That's what all these big global papers are doing, or they're using remote sensing technology to combine with this ground level inventory, or they're doing remote sensing and then using this data to validate remote sensing technology. So this is a paper Andy Finley and Sudipto Banerjee and I worked on, published in the Journal of American Statistical Association. This is Andy's brilliance. I was a minor player, but important thing here is that here are the FAA plots, here is interpolated forest and non-forest area, and here is the resulting biomass map. See what all these black areas? All those blacked out areas are areas where there's no data. They're the urban areas, which we'll have data for soon, and there are other areas that fall into this category of trees outside of forests, which is an area of growing importance. Eventually, we're going to have to do just a wall-to-wall tree census, including all land types. And I hope to be there to help build models for that. So the key idea is that the underlying tree models are critical for anything you want to do with this data at any kind of larger scale, or even at the scale of a plot. And that also that the tree environments vary over complex landscapes. So this is why I want to have a universal tree model that can pretty much work everywhere. So we wrote a paper, our working group wrote a paper in Journal of Forestry, basically a call to improve methods. And we came up with some key issues for doing this work. One is you need lots of data, as I already alluded to. Destructive sampling is costly. My lab, we spend about $1,000 per tree, and we take about 8,000 measurements per tree for destructive sampling. One tree. So put that into your budget. The models they're using, as I showed in the DNR case, they're too simple. They just use diameter of breast height as a predictor variable. So they have tremendous difficulty predicting components. And you need this generality to cover a broad range of species and geographic areas. So you can see why I've been telling them you need universal tree models and move away from this sort of historic regional work that they're doing. Okay, so breaking this down into those three main bullet points of how to improve. Getting better and more data. So basically, in 2011, I was invited to participate in this massive project to develop new national tree biomass equations. 6 other universities are participating, representing different regions of the country. We're building the new tree biomass and biomass component models, and these are going to be compatible with the volume estimation. So think of that formula, volume equals mass times density. If I divide the mass of the tree by the density, I should get the volume. That's what compatible means. If I predict those separately, I'll get 2 different estimates. They're not compatible. So we've been running around destroying trees, measuring the hell out of them. And then we also did something really smart. This was Phil Radke at Virginia Tech's idea. We went around in all the dusty file cabinets and all of the labs around the country that we could find and dug out all these old records, cards from card readers, everything, and created this legacy tree database. So if you're ever looking for tree data, this is a publicly available database. What are we up to? 245,519 tree data records now from all across the United States. I think we threw some Canadian data in there too. So we're solving the data limitation problem. Destroying trees is not nice, as one of my students told me. So what we're doing is we're also trying to figure out non-destructive methods. So here's a terrestrial laser scanner. You basically put this down and you shoot it up at trees, and then lasers come back and you can make really nice 3-dimensional images of forests. This is a student in Harvard Forest. So basically, we teamed up with an NSF-funded research collaboration network of terrestrial laser scanners from all over the world, and we all descended on Harvard Forest just this past August. And so what we did was we used all these different laser scanners and scanned the heck out of these plots. And then we're using what's called quantitative structural models, from which we can take these 3-dimensional images, and then we can actually compute the volume of all the different components. And then led by MSU, here's Sam Clark, we trained all these people to do the necessary destructive sampling to test these detailed models. And we just did a couple of presentations at the Silva Laser Conference just last week in Virginia Tech, but we're not gonna, it's gonna be years before we get all the publications out of this data. Okay, so I can get volume now, I need density. So how do you get density? So MSU hired a new professor in electrical engineering, Jeff Nanzer, who was doing medical microwave tomography to actually scan for tumors. And Emily Huff introduced me to him, and then we started talking, and I said, can we build a band that you can wrap around a tree and do this on a tree and determine the density and whether there's cavities or decay in them? And he said, yeah, I think we could do that. So we got a small amount of funding. We just got this funding, and we're going to try to build this. I keep calling it the Nanzer band. I don't know if he likes that term or not, but I think it's catchy. Okay, so one other method that I looked at was this functional branch analysis. So here we're back to Vinci and fractal scaling and everything. And Meinevan, Nordwick, and Rachmat-Mulia came up with this theoretical model that said, okay, the coefficient of a power function should be related to this fractal branching architecture. So it's a pretty clever idea. And it could be a non-destructive method because what you can do is you can have someone climb up the tree and you can basically measure all of these different diameters at different forks, and then you can just take those measurements, put them into their model. It's a big Excel spreadsheet computer they created, and it spits out the coefficients of the power law. So when I was working on the carbon benefits project in Kenya with David Skoll and company, Shem Koya, who was a PhD student, and I worked on seeing, okay, could we actually do this? And I had the gator eyes. So I was like, oh, well, you don't have to have anyone climb the tree. We can just use the gator eyes and get all these branches. And that part of it didn't quite work out the way I wanted. But basically, we had enough of this information, and we had the destructive sampling. If you're working in the tropical trees, you know, the lasers are interfering with leaves, so you can't always get the measurements of branches. So it's one of the limitations. Anyway, the short of it is, what we did was we had destructive sample trees and trees where we knew those measurements. And so we said, what if we had been able to get them? What would we have done? And so these graphs show 4 different species. This is mango. This is Acacia mearnsii. And these are 2 eucalyptus species. And so what this shows is the actual biomass and the predicted biomass using the functional branch analysis method, the standard allometric equation that is generated, and a 1-to-1 line. And so the short of it is that this method worked well for something like Acacia mearnsii because basically you can look at it. It is kind of a fractal, right? But it worked really badly for the eucalyptuses. And I think this actually in part has to do with the fact that they're growing in plantations. And so you've got this whole trunk length which is messing up the fractal geometry. And also, if you think about it, when trees are competing with other trees, the crowns are banging into each other, so the fractal branching architecture is actually being disrupted. So actually, in this paper, I coined the term disrupted fractals, basically to say, look, trees aren't really fractals. They're disrupted fractals because of all of the injuries and things that happen to them over time. However, because it kind of worked for open-grown trees, and we've got plenty of arborists who could climb trees, I thought this could be a potential method that could be applied for developing data for urban allometric equations, particularly because in urban areas, it's distasteful to people when you come and cut down their tree and ask them to give their trees life for science, like we have been doing with so many of these trees. Okay, building better tree models. One of the gaps was dead tree carbon. So FIA has standing dead trees, and then when a tree leans over and falls down, it becomes coarse woody debris. Now, it turned out that Chris Woodall and some others, including me, looked at this issue and basically said the models that FIA is using to predict dead tree carbon are over-predicting the amount compared to field measurements. That's bad. So we're saying there's more carbon being sequestered in the dead trees than they would by a considerable amount. This is a map from this paper we published, basically showing the distribution. This is field minus modeled, so the red means bigger errors, and the green means smaller errors. But the key issue here was we needed better models. So for those of you who remember him, Hong Suan, who now owns Korea House, by the way, in Trowbridge, did a PhD with me. And he and I worked on this method to improve the abundance of standing dead trees. The key problem is that when you put this little plot in, 1 plot per 6,000 acres, and the odds of a plot containing a dead tree are low. So you basically get what's called zero inflated data. And so we came up with a method where basically, if there was no dead tree in a plot, you would measure the distance to the nearest dead tree outside the plot, and then come up with a model that could get better estimates of standing dead tree abundance. We just recently, our working group, this is the 7-university group plus some FIA scientists, we just published this paper recently saying you can fix the existing models if you just, do a little bit better modeling. Just use more sophisticated models with the same data and approaches, which people are happy about. I'm advocating, though, that, let's not take the old car and keep putting money into it. Let's just go with completely new models. So I've proposed some new models. So basically, getting back to volume, mass equals density times volume, we've got these nice stem taper models. And so there was already a method where if you knew how the density of the tree changed from top to bottom, you could integrate that and get stem biomass. Well, Neil and I already developed a way that you could integrate the volume of the whole above ground tree. So we published another paper basically saying you could do this for not just the stem, but for the whole tree. Call these vertically integrated whole tree biomass models. So the way that works is you take our volume profiles I showed you earlier, you multiply by the density profile. So this is the density from the bottom to the top of the tree. You see there's some crazy patterns in there, which makes it hard to model. And then there's different lines are for the stem versus the branches. So the stem and the branches don't have the same density, and they also vary differently in the vertical structure of the tree. So it's not as simple as multiplying them, but basically we get these profiles. And what we wanted to test is what if you didn't have this density profile, but you just had like a constant density. You just said this is the species average density for that individual based on published values. Because density is hard to get. These two different functions, the constant means assuming a constant, and the solid line is integral. And so overall, obviously, you can get not so bad predictions with both, but you get some really, you get better predictions if you use this integral. Now, these density profiles are hard to get, so I said, what if we simplify further? So this is the next iteration model. Basically said, okay, a tree is simply the sum of all of its parts. And so we estimate mass for each of the parts using these models. And these models look basically at the dimensions of the part, the diameter squared of the tree, and the density. And then you have this form factor, which relates the form of the tree to the measurements. Okay. The cool thing about this is you can predict the mass of all the components, and then you can, like I did with Einstein's equation, you could just reverse it, and you can predict the volume. Now, everybody likes volume because volume is more tightly related to commercialized production of the tree. It's something we know how, volume, how much dollars per volume it is, much more so than mass. But if they did adopt this approach, they would have to concede something on the accuracy of the volume prediction. Anyway, I'm going to the national stakeholders meeting next week, along with Neil, Andy, and Garrett Detman. And I'm going to discuss why I think that this would be a good trade-off for them. The model works extremely well for all the species, hardwood species I piled into it. I didn't have conifer data at the time that this paper was published, but we're fitting it with a much larger data set now. It gives us great estimates of all the components. And then the nice thing about this is you can just add up any set of pieces of components to get the part of the tree that you want. You want the mass of the stem, it's the sum of these three components. You want the mass of the crown, it's the branches plus the stem part that's inside the crown. See how this is cut out from there? And so on. You get the whole tree mass. And you can work all these things in reverse. So you should be able to predict the stem from the crown. So if now I have some remote sensing technology that can predict the crowns, right? We can't see the bottoms. I should be able to predict all the other parts from that. So really good, 75% reduction in error over an added model where you use only DBH. So you can use diameter breast height to predict all these components and add them up, but you just get much less accuracy. And the practical issue is getting the density, right? So for these trees, we knew the density of every tree because we destructively sampled it. Well, how are you going to get that? Well, with the Nanzer band. No, you can't get that repeatedly. So one of the things people do is they use published values, species average values. So what I did is I reran the model and said, what if we only had the published values instead? Still get a 57% reduction in the mean squared error. So most of the improvement is coming from the improved volume estimation, not from the density estimation. Great model. Will it work across many species and locations? So I tested this with Michigan data. So I started thinking about, okay, you've got the genetic code of a species. So inside every individual tree, there is a plan in its genes that says, grow this way. But then there's all these disruptions to that growth pattern. And those are coming from the environment. And the main ones come from competition from other trees, shading, and environmental factors, such as wind. The tree has to build an architecture that's biomechanically stable, or it breaks apart. We've had, unfortunately, many tests of this in the recent months. So I just published this paper in Functional Ecology with Brian Kane of the University of Massachusetts, where we looked from open-grown trees to trees that are crowded and looked at these trade-offs in tree architecture, balancing crown competitives with wind resistance. So it was very interesting from a tree biological perspective. So these are your typical crown classes. FIA already measures these, but doesn't do anything with these classes. They just put the trees into classes and summarize the data. What I'm showing here is that you get different allometric relationships depending which crown class that the trees are in. And you get all kinds of different branching architectures. I came up with a bunch of weird branch metrics because I had all that destructive sampling data. Like this is average branch slenderness. This is branch mass inequality. These are all ways to describe the crown in different ways from a mass allocation perspective. All of them are affected by the canopy position of the tree. And this is the wind resistance. So open-grown trees have the highest wind resistance, and it exponentially declines towards overtop trees in the understory. They're shaded from the wind by all the trees that are crowding them out. So they're losing the light competition battle. They don't add allocate a lot of mass to protecting them from the wind. That's why if you open up stands, some of them will just fall over. That's because if they're planted too dense, they were shielded from the wind. They don't allocate a lot of structural mass to avoiding breakage. So a lot of urban trees are open-grown trees, so we wanted to look at whether there was urban trees could be modeled pretty much as open-grown trees. And I'll basically tell you the answer, and you can read it in that paper, is the key difference for urban trees is fewer tree neighbors. So if you want to create a continuous field of tree model, basically if you model the neighborhood of the trees around the trees, then you capture most of the differences in the allometric relationships from open-grown trees to trees that are crowded by other trees. So I tend to combine this generalized tree modeling with both terrestrial laser scanning and above canopy LiDAR, basically build a continuous field tree model. So the next steps are these universal trans-Pacific models. If we want to do this for the entire United States, there's a lot of species. And even with the work we've been doing with thousands and thousands of records, you still have huge gaps. So we're using much larger data sets now. I'm going to apply these models and building really functional trait-based models. Garrett Detman's master's thesis is a really nice model that uses functional traits. And what this enables you to do is predict for a species that you have no data for, because you've developed an all-species model, or what I'm calling a trans-specific model. Look here, this is the mass of the branches, and this is the mass of the stem. So we could look at that relationship for trees in different canopy positions, open grown, and different tolerance classes, and see how those relationships change. Even with, so I had 4,318 trees to produce this graphic. And I still have, look, there's no tolerant open-grown trees in the entire database. So that would be like actually, when a windstorm hit MSU, I had MSU grounds cut up some firs, some Nordman firs that fell down. So we actually now have some data for that. But all right, I think I'm probably at around 55 minutes. Sorry it was a long talk, but I had to talk about like 8 years worth of work. So a lot of people have contributed to this. Almost everything I showed you was somehow contributed by a lot of different people. So thanks.